Hey guys, so welcome back. This video is part one of a two-part video series entitled Biochemical Techniques and Tools, in which my goal is to try to provide you with a bird's eye view of how a biochemist might identify, express, extract, purify, and analyze a protein of interest, say maybe to better understand a biochemical process in the cell, or possibly develop some new drugs or therapeutic strategies in order to treat a novel disease. Part one of this video is going to focus on the front end in terms of expressing, extracting, and purifying a protein of interest, where part two is going to focus on the techniques to analyze and characterize that protein. So at the end of part one, when we meet in class, we're going to work on section 5 from your workbook, in which you're going to be able to get some practice in describing the molecular basis for some of these techniques. So how do these techniques work and why do they work? As well as giving us some practice in how to properly select techniques or a sequence of techniques in order to accomplish a given isolation or biomolecular characterization objective. And it will also give us some practice in utilizing our critical thinking and problem-solving skills to then identify the necessary tools that we need in order to accomplish these given objectives. All right, so again, this is just part one of the video series. Do follow this up with part two, but we are going to break this up into two class meetings. So class meeting 1, we're going to focus on S5 from the workbook. Class meeting 2 is going to focus on section 6 from the workbook, which is a little more of the analysis of our protein. In our class for section 5, do try to bring some laptops or devices with you. There's a lot of animations that we'll be working with as we go through this activity. All right, so I will see you guys in class, and let's get started with the lecture.
Hi everybody. Today we are going to be talking about techniques, methods, analytical methods, tools that biochemists use in the lab on a relatively daily basis. So in order for us to analyze it, I thought the best way to kind of go over various biochemical techniques is to sort of kind of go over a mock expression, purification, and characterization of a novel enzyme, Verbosase. So this recently discovered enzyme is found in extremely high concentrations in overly loquacious professors and politicians, and however is found in extremely low concentrations in very quiet and shy students. So little is known about this relatively recently discovered protein, except that we do know that it's 173 amino acids long, that it is only active in the presence of calcium, and it contains a number of glutamate or aspartic acid residues. And then it weighs approximately 19 kilodaltons. 19 kilodaltons is the equivalent of 19,000 grams per mole. Commonly when we talk about proteins are discussed in terms of daltons, D, or kilodaltons, which is 1,000 daltons. All right, but 1 dalton is the equivalent of 1 gram per mole. So it's a molecular weight. So the first thing we need to do to study verbosase is to express it. Now biochemists routinely grow proteins up by expressing them or letting cells, such as E. coli cells, grow the protein for them. So y'all may have remembered in class I mentioned that synthetically making proteins is very difficult because when you make, say, this protein is 173 amino acids long. If we make a polypeptide that's 173 amino acids long and we just synthesize it, well, that can fold in so many different possibilities. I mean, the possibilities of it folding are endless. to the point that usually the only way that we can successfully synthesize a protein is to synthesize it using a cell. So the first thing we have to do in order to synthesize verbosase is to identify the DNA sequence. Luckily, the DNA sequence has already been identified to us by previous researchers, and that data has been published. So a lot of times they will utilize agarose, gel electrophoresis, and enzymes to determine the sequence of a gene. If you want to learn more specifics about determining the sequence of a gene, you can follow the link here. I don't personally have too much experience with it. I understand the theory behind it. This lecture is really just to kind of give you a survey of the various tools that we use. So I'm not going to try to go too much in depth on how we sequence a gene, because really that could be a whole week of lectures in and of itself. But do know that to sequence the gene, we utilize gel electrophoresis with agarose gels, enzymes that cleave at specific nucleotides, and then we can run the fragmented DNA sequence, and where we see the band show up, we can determine the sequence. Okay? So, let's go over this. So let's say we had an enzyme that cleaves at every adenine. We will have fragments that end at an adenine. So going from our longest fragment, our shortest fragment to our longest fragment, we can figure out where there's an A in the DNA sequence. Here we have an enzyme that cleaves at every thymine. Here we have an enzyme that cleaves at every cytosine, and an enzyme that cleaves at every guanine. So starting from our shortest fragments, A, T, A. Oh, there's a C, T, C, A, so on and so forth. So this link right here is a great little tutorial. It has some practice for you, kind of walks you through how a gene is sequenced. So once we know the sequence, we can then take the sequence to verbosase and insert it into a vector or a plasmid, which is a circular piece of DNA. This plasmid already has a promoter region as well as a code for an antibiotic, and we'll talk about why that is important in just the beginning. So then, once we have our plasmid that's inserted with the gene for verbosase, we're going to insert that plasmid into a host cell. Commonly, E. coli is used because they're easy to grow up, also because E. coli can be killed with antibiotics. So now we have a way of determining if the plasmid indeed was inserted into the E. coli cell. Remember, this plasmid can code for antibiotic resistance in these E. coli. So E. coli that have the plasmid will survive antibiotic treatment, where E. coli that did not get the plasmid inserted will die. This way we're growing up specifically E. coli that will express our protein. Now we've inserted our plasmid, we've treated them with antibiotics, we've killed off any E. coli that do not have the plasmid inside it, and now we're just going to let our cells sit in a flask similar to this, usually at about 37 degrees C, and shake. So we're going to shake until we've reached optimal cell growth. Once we've reached an optimal cell growth, then we are going to turn on our gene by inserting or adding a molecule to the solution that will activate our promoter region. So we add some sort of activating agent that comes in, binds to the promoter, and then we are able to start the Verbosase messenger RNA will be transcribed, sent to the ribosome, and now our proteins are going to start expressing or growing the verbose protein. Now, because we have a ton of cells in here, and this promoter region is going to be constantly turned on, these cells are going to produce lots and lots and lots of our protein inside the cell. Once we've allowed that to happen, so we've got so here's our cell, and there's just lots and lots and lots of our protein, our verbosase. All right? Once we've done that, we need to collect our cells. So we may have vessels or flasks that have liters upon liters of solution with some cells in them. So we spin the cells down using a centrifuge so that we get a pellet. Then we can pour out the supernatant or all that buffer that the cells are living in and get rid of it because we don't need it. And now we just have our pellet. So now that we have this pellet of cells, our protein is trapped inside the cells because our cells have plasma membranes. So we need to break open the plasma membranes of these cells so that it can release our verbosase protein into solution. So normally we resolvate our pellet in a minimal amount of buffer. We can either use a French press to force the cells to break open, or we can use cell lysis buffers. So these are buffers that contain detergents, lipase, other molecules that can break open the plasma membrane. We can then recentrifuge so that the insoluble hydrophobic biomolecules, such as bits of the plasma membrane and other membranes and other fats and anything else that's hydrophobic and insoluble in water, will separate to the bottom, so you'll get another pellet from the aqueous layer that contains hydrophilic molecules. So now we have two layers. We have hydrophilic aqueous molecules on the top, liquid, and then we have a hard pellet of hydrophobic molecules. Now, because we know verbosase is globular protein, and remember I said it had a lot of aspartic and glutamic residues, so it's got a lot of negative charges on it. We know that this enzyme is soluble in the aqueous layer or in water. So for this reason, we know that our protein is most likely in the aqueous layer and not in the pellet. However, if I was a good biochemist, I would still keep the pellet and test it just in case I finish analyzing the aqueous layer and the protein isn't there. I still have the pellet that most likely my protein is in. Okay? It's got to be in one of two places. Now, to purify our target protein, a commonly used technique by biochemists is chromatography. If you remember from organic chemistry, we talked a lot about chromatography. You even did a little chromatography. But basically what chromatography does is it separates molecules based on some sort of physical property. So for this instance, we're using it to purify our target protein from a soup of other globular proteins that are found in the aqueous layer, as well as other biomolecules such as nucleic acids or some water-soluble sugars and some other molecules from the aqueous layer of the cell lysate. So for instance, we have a mixture, and in chromatography you have what's called your stationary phase. So we'll kind of make a little stationary phase. Usually it's a resin, but I'm going to make, for this instance, a resin that has a lot of hydroxyl groups, such as in silica. Okay? So we have a lot of silica. It's a stationary phase. It's solid. It does not dissolve in our mobile phase, which is some sort of liquid solvent that runs across or down the stationary phase. Now, normally, your mobile phase or your solvent is going to be an opposite to your stationary phase. So if our stationary phase with all these hydroxyl groups is very hydrophilic, our mobile phase may be hydrophobic or nonpolar. Maybe in this instance it's hexane. Okay? So in this mixture, molecules are going to separate based on their interaction between the mobile phase or the stationary phase. So if a molecule is very hydrophobic, it's not going to interact very much with the stationary phase, and it's going to flush through the system and just go through with the mobile phase. As you can see here with this blue fraction, it's the first to come off the column, so I would predict that it is the most hydrophobic and does not want to interact with the stationary phase. Now our next fraction, this red fraction, may be amphiphilic. Maybe it has some hydrophobic functional groups as well as some hydrophilic functional groups, so that it interacts both, but it doesn't really have a preference for one or the other, and so it kind of comes out in the middle, and we can collect that fraction. Now our green fraction may be slightly more hydrophilic, so it's going to interact more with the stationary phase than it did with the mobile phase, so it came off last. Now when we're talking about proteins, we can separate on a number of features. So to give you another analogy of how chromatography can be used to separate compounds, I like to think of, an analogy comparing how we can separate students or determine students based on their eating habits. So let's say we have cafeteria, Subway, anything else, a bar. So now we have a mixture of students, and they're all different years. So we got a mixture of third year, fourth year, first year, second year students. So as our students are going down the pathway of various eating spots, most of the time your first year or your freshman students are going to stop at the cafeteria. Now your second year students are going to move on a little further because they're tired of the cafeteria. So they may stop at Subway. Subway's still on campus, but hey, it's different. It's something other than the cafeteria. Our third year students haven't eaten at the cafeteria for a year and now have eaten at nothing but Subway for a year, will go anywhere else they can go. Anywhere. And our fourth year or our senior students have just given up and they're at the bar. So I hope that was a good analogy and will help you understand kind of how chromatography works. If not, we can talk about it more. So now I said, you remember there's different types of chromatography. So we can separate, again, by hydrophobic interaction. So this utilizes either normal phase or reverse phase chromatography. So in normal phase, we have a silica-based stationary phase that's got lots of hydrophilic residues with hydroxyl groups, oxygen, silica. So hydrophilic molecules or polar molecules are going to love to interact with this stationary phase, where hydrophobic molecules are going to pass on through. Now in reverse phase, we have the opposite. So a lot of times in reverse phase, the silica gets modified with carbon-hydrogen chains to now make this mobile phase nonpolar. So now nonpolar hydrophobic molecules will interact with this silica, while hydrophilic molecules will move on by. We can also have ion exchange chromatography. This works when your stationary phase either has formal positive charges, so proteins that have negative charges will stick to the stationary phase, where proteins with positive charges will flow on through. If we had a negative stationary phase, then positive charged amino acids would stick, while the negative ones will go on through. To elute your target protein from the stationary phase, you have to change the charge of the protein. So think about, how can you change the charge of an amino acid? So that's one way to remove the protein that you want to purify. So once you've adhered it, you've washed all the unwanted stuff through, now you need to recollect your target protein. How do you do that? Well, one way, like I said, is to change the charge of the amino acid. And we'll talk about how you can do that. If you haven't come up with the answer yet, we will talk about that in class. Another way is you can flood the system with positive charged ions. So we could flood it with sodium salt. So then the sodium salt that's flushing through will compete with the stationary phase, and eventually you'll wash all your protein away. So there's two ways to do it. Another type of chromatography is affinity chromatography. Here our stationary phase has a molecule that is a ligand for your protein for verbosase. Remember, verbosase binds calcium. So we could put calcium molecules on our stationary phase, and then any protein that binds calcium will bind to the stationary phase. So as you can see here, the target protein binds to the stationary phase, and the rest of the proteins, the unwanted proteins, are washed away. And now to elute or to recover our protein of target, you usually flush the system with the ligand. So in this instance, we would flush with a buffer that has lots of calcium, and that way it will compete the protein off of the stationary phase. Another type of chromatography is size exclusion or gel filtration. This works, your stationary phase is filled with beads that have holes or pores in the beads, and these can change based on what size protein you're looking at, and what you end up with is you separate proteins based on their size. So depending on the size of the pores, molecules that can fit into the pores will get stuck and will filter through more slowly through the beads, while molecules or proteins that are too big to fit through the pores will just go around the beads and flow through faster. So as you see, large molecules will flow through first, then smaller molecules will flow through last because they're coming in and out of the pores of the beads. This is usually used as a final purification. It's a really, really good purification technique. It just often takes a long time to do.
Hi everybody. So just to recap, you just finished part 1 of the 2-part video lecture series, Biochemical Techniques and Tools. So in part 1, we had looked at how a biochemist may express and then extract and purify a protein of interest in order to better understand maybe a novel biochemical process in the cell, or to develop some novel therapeutics or drugs that might target this protein. Part 2 of the video lecture series is now going to look at how we characterize and analyze the protein that we just purified. Please again remember to bring a device, a laptop or a tablet to class with you when we work on section 5 from our workbook. Okay, I will see y'all then.